Once all the iPSC colonies are detached using DISPACE, we're going to go ahead and take them to the biosafety cabinet, and we're going to go ahead and neutralize the DISPACE. We're going to be using DMEM-F12 to neutralize the DISPACE. Use 2 milliliters of this basal media. And remember, this basal media was pre-warmed prior to using it. We're adding 2 milliliters to each well on both 6 well cell culture plates. Make sure you mix it. And now we're going to go ahead and collect all the iPSC colonies and transfer them to a 50 ml conical tube. When you're transferring the cells, just be very careful not to break any of the colonies. So be careful when you are pipetting up and down. Make sure you reduce the speed of the pipette gun prior to starting to ensure that you dispense the cell solution at a drip. By doing this, you're just preventing breaking any of the colonies. If you dispense the liquid too quickly, you will end up breaking the colonies and you'll have a harder time forming embryoid bodies. Just a reminder, these iPSCs were cultured in a feeder-dependent system. So the iPSCs were cultured on MEF cells. And once the iPSCs reached a higher confluency, that's when we use DISPACE to detach the cells, and the iPSCs are now ready to be cultured to be formed into embryoid bodies. And then when we form them into embryoid bodies, that's when we are going to be differentiating the iPSCs into cardiomyocytes. So the reason why we use a feeder-dependent system is because the cells grow more efficiently and a little bit faster than if we were to grow them in a feeder-independent system. Now that all the cells are collected, all the cells will now just settle towards the bottom of the conical tube. So if you notice, we're actually not going to be vacuuming up the spent media. We are going to be pipetting the media out of the conical tube. So at this point, all the iPSC colonies have already settled to the bottom of the conical tube, which means that when we take up the media using the pipette, we won't be taking up any of the iPSCs. So if you notice, right now what she is doing is actually rinsing off any of the iPSC colonies that might have attached themselves to the inside of the glass serological pipette. And she's trying to rinse them out to potentially rescue any of these iPSC colonies. Be very careful not to take up any of the iPSC colonies the closer you get to the bottom of the conical tube. So now we are going to be using a new serological pipette to add DMEM-F12 to the cells So we're going to be using 20 milliliters of DMEMF12 to rinse the cells. This just allows all the disc paste to be taken out of the cells. Because again, we are not centrifuging these cells. If you were to do that, the cells would just clump up and you wouldn't be able to plate them properly to create embryoid bodies. So now just let the cells settle. Once they settle again, you'll be rinsing the cells again. So right now, we are trying to rescue any of the iPSC colonies that were initially stuck to the inside of the glass serological pipette. So you'll just take these out and try to collect them into one well. These will later be added to the conical tube. Once you've rescued all the iPSCs, you can go ahead and vacuum off the rest of the spent media. Again, remember, these do not contain any of the iPSCs. Now she's just collecting the rescued iPSC colonies so that she can go ahead and add them to the conical tube. She got rid of the spent media first and then collected the colonies. Let those settle and vacuum off the rest of the spent media. So now that the colonies have settled, you can go ahead and vacuum off the media. Just be very careful when you're doing this because you could potentially take up some of the cells that haven't settled properly. So just be very gentle when you do this. Be sure not to move the conical tube because you could potentially lift up some of the iPSC colonies. So you're gonna wanna rinse the iPSC colonies a total of 5 times. Again, we need to rinse more just because we are not centrifuging the cells, which means that we're not taking up all of the spent media, which means that there could potentially be a trace of disc paste. So by rinsing 5 times, we're ensuring that we're really getting rid of all the disc paste from the cells. So now we're gonna go ahead and add the cells to an ultra low attachment plate because again, these cells are going to be cultured in suspension. We do not want these iPSCs to attach the plate during the first 2 days. So this was the last wash. I went ahead and fast forwarded through it. So just remove the rest of the media and those will be ready to be plated. So this right here, this is your hiPSC-TnnI1-MEF media that does not contain basic FGF, but does contain the ascorbic acid. So you're gonna wanna add 10 mils of it to your cells. You can go ahead and add it to the side of the conical tube so that you do not break up the colonies. Take up some of the cells. Again, do not pipette up and down too much. If you notice that the cells aren't really mixed well in the media, go ahead and just give it a swirl and then take up your cells. You're going to be adding 3 mils of cell solution to each well. Once you've plated all of your iPSC colonies, you are done. That is all you need to do. These cells will be incubated for the first 2 days in an ultra-low attachment plate using that specific media. You could see the colonies all in the plate. Make sure you visualize them underneath the microscope. And you're going to be incubating these cells in the 37-degree incubator, and that's how they look.